Okay, we're going to get started. Thank you all for coming. We have a lot of presentations, which is in part a problem and in part just a reflection of how busy we were at the ACC. There were up to 7 wonderful late breakers presented at ACC. We're going to do 4 of them today, time allowing, and we're going to cover them in rapid order. So starting us off, we have the Artemis study given by Tracy Wong. We're going to have Indy given by Adrian. We have Treat given by Chris Granger and Secure given by Renato in the end of the day. Each presenter will present for about 5 to 7 minutes. We'll have them come up here and take a question or two from the audience, and then we'll have a round table at the end if we have time. So beginning us will be Tracy, who will be presenting Artemis in fine fashion.
Thank you. Sorry guys, I forgot to dress up. So where's the slide advancer? Who's missing a tie in the audience? All right. So I'm talking about Artemis, and just to set the stage for Artemis, I'm going to show you some slides that of data that we've collected from about 11,000 patients that we enrolled in Artemis about their medication taking behavior. So one of the first questions was, and perhaps you want to ask yourself this question if you take your own medications at home. In the last month, how often have you missed taking a dose of your own medication? And as you can see on the slide, more than half of the patients admitted to us that they missed taking a dose of their medications at home. Second question, how important is any of the following things to the fact that you need to take a medication or that you choose to take a medication? And I highlighted, sorry about the formatting, we lost the labeling there. I highlighted one column, which is 47% of patients thought cost was the key thing to choosing a medication. So we know patients don't take their medications well. We know that cost is really an issue here as well. We went on and probed some more. Financial hardship. How often is taking a medication difficult for you financially? And you can see, again, more than half of the patients said that there is some financial hardship, and then we pushed them. Have you ever not filled a prescription before because cost was a problem for you? So someone took the trouble to send a prescription to a pharmacy, and the patient went to the pharmacy or chose not to go to the pharmacy because they knew it was going to be too expensive and they couldn't afford it, and that was 17%. And this is a slide that I had put together for another talk previously, which basically shows some of the problems that we're dealing with here. And this is real. Out of every 100 prescriptions that are written, about 50% are picked up from the pharmacy. About 25% of these are taken properly, and at the end of the day, only about 15% are refilled properly. So we have a big problem with our patients taking medications. So what did we do in Artemis? So in Artemis, we had the following hypothesis. We basically said that by reducing and equalizing the out-of-pocket cost for patients when they're picking up a particular class of therapy, P2Y12 inhibitor therapies, and there are generic versions of that and brand versions of that. But by reducing that cost, equalizing between the brand and the generic, we hypothesized that the treatment choice for me as a clinician to the patient would be dictated more by the patient than what the patient could afford. So if I felt the patient really needed it, I'd be able to prescribe them the drug that they needed. 2, we felt that patients now would be able to take the medication for as long as they're supposed to be taking the medication, so what guidelines tell us to do, which is a year of treatment with this particular class of therapy. And that 3, as a result of both, we would actually make patients feel better, keep them out of the hospital. So we cluster randomized hospitals. So we recruited about 301 hospitals across the United States. About half of these hospitals were randomized to the intervention arm. The other half were randomized to the usual care arm. Usual care arm just treat the patients as they usually do. In both of these arms, the treatment choice was totally up to the physician. But in the intervention arm, these hospitals were able to discharge their patients home with a study voucher that helped the patient pay for the medication that the physician prescribed for them. So what do we see here? Well, we saw an 18% decrease in the amount of clopidogrel being used in the intervention arm, which basically tells us clopidogrel is the generic version of the drug. The brand versions are the ticagrelor and the prasugrel. It tells us that because we took cost out of the picture, clinicians were more likely to use in some cases, more effective medication to be able to treat the patient. So that's great. What else do we see? Well, the second hypothesis was that patients would stay on treatment for longer. And again, we see that here as well. So the interventions arms in the green, the usual care arms in the white, and we see that patients who are in the intervention arm are less likely to prematurely stop their medication. Okay. Finally, let's look at clinical outcomes. And here we had a big surprise. We did not see a significant difference in the outcomes. And so we had to ask ourselves, why is that? Well, part of it may have to do with the fact that we're only dealing with one type of medication. And a post-MI patient needs to leave the hospital with maybe 5 medications, if not more. But also, we had to do it by a voucher, meaning, we've got different patients being treated with, covered by different insurance plans, so we needed to be able to treat all of these patients, and the way we could do it was by handing them that voucher. But about 28% of these patients never used that voucher. So if we do an as-treated analysis, where we looked at the patients who did use the voucher versus the usual care, now we start seeing a little bit more divergence in those clinical outcomes. It was 7.5% versus 10.6%. So possibly if everyone was who got the intervention actually used the intervention, we might have seen a different case there. So at the end of the day, we conclude that a copayment reduction strategy is helpful to us as clinicians so that we can do the right thing for the patient. It also helps us conclude that a copayment reduction is helpful to the patient. They can stay on the treatment for as long as they're supposed to stay on the treatment. But if we want to really think about improving patient outcomes, we may need to go beyond copayment reduction alone as a single intervention here. And I put on this slide here the many, many, many people here at DCRI who really helped us get Artemis to the finish line, and a huge thank you to you all.
Great. Up. Take a seat. Up.
I can't eat my cookie.
So, questions from the audience. Chris Granger. Shocking.
Important cluster randomized study. With respect to the issue of course, the issue about patients who use the voucher having better outcomes is pretty confounded. Because people who adhere to things, even placebo, have better outcomes. But did you see that? Did you see, for example, did a person who used the voucher, was they also more adherent to other medicines unrelated to the voucher?
We're doing that right now.
Okay.
So I don't have the answer to that right now.
My prediction, they will be.
From the patient adherence standpoint, we saw about a 16% relative increase. But in terms of an absolute increase, it was about 3%. And part of it is because you ask a patient, are you taking your medications? They probably are going to tell you that they are taking their medication. So overall, the adherence rate there was somewhere around 85%. So 85% of our patients said, I'm taking my meds. The interesting thing about the study is we decided not to take that at face value, so we also did a couple of things. We went to the pharmacies that the patients filled at to get their fill records and looked at their persistence that way. And we saw a very different set of numbers. So there it's 55% of patients, almost 30% difference here. So 55% of our patients are actually sticking to their therapy. And now when we look at the relative difference, again, we're now seeing a bigger difference. So we're now seeing up to about 30% or 35% difference between the intervention arms. So the good news is that we're seeing the same direction of effect. In terms of the magnitude of effect, I think it might in part depend on how we're measuring that particular outcome.
Great. Stay up there. We're going to come up with... You can't move, actually. Chris, I think you're up for next. Or Adrian?
Who's next?
Adrian.
Next slide.
Sorry.
The next slide.
Next slide. That would be me. Sorry. Indy.
I'm happy to present it if you would like.
Okay, so with a show of hands, let's see who likes beets. Beets. Have you ever thought about smoking beets? Inhaling beets? Okay, Well, good. So we did the right study. So we tested something really novel, which is essentially understanding nitrite delivery to improve exercise capacity in patients with heart failure and preserved ejection fraction. I'll get to the beat story here shortly. So we know patients with heart failure are short of breath. Patients who have preserved ejection fraction, this is one of their main problems, is having difficulty doing things every day due to shortness of breath and their exercise capacity. There have been interesting basic science studies looking at nitric oxide and its signaling playing a role in pathophysiology. And one of the things that has come up in the Heart Failure Network is we actually tested one example of that, which was with another form of nitric oxide, or at least isodyl dinitrate or mononitrate, and really did not show any improvement in actually activity levels. So this is one of the first studies that actually used activity before Fitbits were cool. And that was called the NEAT study that was published several years ago from the Heart Failure Network. So the question that arose from that, well, do we have the right mechanism in terms of delivering or improving nitric oxide? And organic nitrites may be the answer. The most common way that people are exposed to that every day is through beets. In this case, we were actually testing a novel medical product that would essentially have inhaled nitrites given 4 times a day, potentially. In our study, we did it 3 times a day to see whether this improved exercise capacity in patients with heart failure and preserved ejection fraction. So you could either inhale this or you could eat a bunch of beets. We chose to try to do the inhalation method. And the population was a typical population with heart failure and preserved ejection fraction that had symptoms, that have objective evidence of heart failure, and they had to have reduced exercise capacity done by a cardiopulmonary exercise test. And then also importantly to have symptoms related to heart failure that we could potentially show an improvement. This is an interesting study design. It was similar to what had been done in the Heart Failure Network before with NEAT, which was a double-blind, placebo-controlled crossover study. Patients would be identified. They would go through a screening evaluation, and then they would get a test dose run-in, and if they tolerate that, then they would go on to randomization. They would go initially no drug for 2 weeks, and then one group would be randomized to nitrite inhaled 3 times a day, and the other would go with placebo, and there's titration up there. And then we would do a CPX test and other secondary endpoints, and then we would go through a washout phase and then crossover. So relatively unique design here, and then we'd end up with repeating all the endpoints again. So our primary outcome was exercise capacity with a peak VO2. And then we had a number of other secondary endpoints, and the ones that people are really fascinated with these days are activity measures with an accelerometer. And then we also had a number of other heart failure standard endpoints, such as quality of life, BNP values, and echo and imaging. The population here is pretty typical for a heart failure and preserved ejection fraction, age of 68. The study itself was just over 100 people, so there are some numerical imbalances across different categories, but largely represent what we typically see here. And again, similar features. They did have exercise impairment with a peak VO2 of 14, and just to kind of give you a frame of reference, like a group that's in their 60s, probably would have a peak VO2 of 20. If you're a cyclist like Lance Armstrong at his very peak level, especially with enhancements, he can get close to 80 or so. Unfortunately, we found out that inhaling nitrates did not do much. So the question is still out there for beets, though, which there is an ongoing study being done at Penn. And there was no difference between placebo or inorganic nitrite, and despite us questioning Steve McNulty over and over, did he make sure he got the assignments right, the p-value was 0.27, and so really remarkably no difference. The one thing about nitrites that are inhaled, they have actually a relatively short half-life. And so while the preliminary studies had shown that you can change hemodynamics acutely, that doesn't translate into something sustained. And so we think this is one reason why we showed no difference. We also were very clean and showing no differences in any of the other outcomes. And so I'll just say for DCRI itself, this presents a grand opportunity for doing studies in heart failure and preserved ejection fraction because we don't have the right therapy yet. And I'll give a plug to the SPIRIT study, which will, I'm sure, be enrolling like gangbusters very soon. So in summary, this intervention did not really change anything in terms of exercise and other measures of heart failure and preserved ejection fraction. It does offer a repeatable or reusable design to get early assessments of whether something has potential for something that's patient-centered in terms of symptoms or functional outcomes. And so we think that this is actually something that can be repeated, relatively small studies to give proof of concept here. And we are back to the drawing board for this condition. So Duke was also a center here, and DCRI is the coordinating center, and here are the friends and families of the Heart Failure Network. I'll stop there.
Great. Questions for Adrian? Chris?
So I accept that I accept your conclusion, but let me just challenge you a little bit on it. How do you know this is a model that works? If there was, seems like to know a model works, you would like to see that a treatment that improves outcome based on kind of some other gold standard or something improves outcome in this model.
Well, so I guess two things. So certainly the kind of leap from kind of a improvement in functional status or exercise capacity does not necessarily mean a change in clinical outcomes. So I think that's one thing just to distinguish. So in terms of the study saying evaluating can someone have a functional improvement, I think you can get a lot of data with these types of study designs to show whether there is actually improvement in functional capacity or activity level. Now, the question we often raise is, do we have the right population? Do we have the right drug? Do we have the right dose? Do we have the right exposure? Do we measure the outcome the right way? And here, while one could go and say, well, we didn't maybe get the right exposure because it does have a short half-life, we may not know if there's something that has a longer duration has a better outcome. So.
And presumably you can look at some patients probably did have some improvement irregardless of the treatment. Mm-hmm. And then maybe you can see if there's correlation between improvements in some of these measures and symptoms and biomarkers, and presumably you'll do those things.
Right, so like to look to see if there's responders. So like, I mean, it's a relatively small study, so it's kind of hard to really assess that because we did look at that, so just on the other end, you can see some responders, but then on the other hand...
Well, I wasn't so much thinking of responders, I was thinking of correlation of change. Mm-hmm. They probably may have nothing to do with the treatment, but some people probably changed as a natural course of what happens to people, and to see whether there's correlation between various measures might be interesting.
Right now, so.
Other questions? Yes, Marty.
Are there any learnings operationally as far as getting sites for CPAT, to get patients to the 2 visits for CPAT?
Yeah, not to do it, but no.
We're doing a site feasibility right now. That's why I'm asking.
Yeah, so I mean, I think so we've done these types of studies in this population with NEAT and RELAX, and so do you have to be pretty selective in terms of the study centers that can do CPAT. The other thing is that we're hoping that we can transition to these types of studies instead of doing CPAT to doing things that are more passive measurements such as activity level. Because then we're not requiring people to come in-house to do these testing. If you've ever seen one where you're wearing like a scuba gear, essentially, it's not exactly the most comfortable thing, but And so those are things, but it does take special sites.
Great.
Okay.
Okay. Can join him up on the stage. Next we have Chris Granger.
Yeah, well, you're going to block the.
Okay, there you go. Sorry. We'll bring him back up.
So my first disclosure is actually Renato should be presenting this, but he graciously, rather than doing two, since I was somewhat involved, but less involved than Renato, and this really is a Brazilian study run by Otávio Berwanger, who many of you know, who's a close friend and colleague and research colleague of Renato's, and a key part of our kind of Brazilian connection. And Adrian, this is another trial that showed no difference, or at least not a lot of difference, between two treatments, and that I think still has some implications, although you can certainly challenge me, as others have, about what the impact of this should be. But I think it does. I think it will change guidelines, and we still are waiting 1 year follow-up. This I'll just show you what was presented and published at the ACC on the primary outcome, which was the 30-day outcome looking at bleeding. And the trial was looking at ticagrelor versus clopidogrel after patients with ST elevation myocardial infarction treated with fibrinolytic therapy. You could say, why is that important? Because we don't use much fibrinolytic therapy anymore. But actually around the world, the predominant treatment to improve survival with reperfusion for patients with ST segment elevation MI is fibrinolytic therapy. So I think this actually does have important implications for global treatment. And even in North Carolina, about 10% of our patients with STEMI in North Carolina are treated in places where they simply can't get to a cath lab fast enough, and they're treated with fibrinolytic therapy. And then we wonder what to do with them when they show up at the Duke cath lab or at a cath lab. We wonder what to do with them. Is it okay to go ahead and use ticagrelor in the first 24 hours? And the other key background to the trial is the Plato trial, which was run by Lars Valentin, and with which we had a role, showed that ticagrelor is better than clopidogrel at reducing cardiovascular death, MI, and stroke, and in fact also improving all-cause mortality. But importantly, there was a gap in this trial in that patients who'd received fibrinolytic therapy for ST elevation MI within 24 hours were excluded from the trial. So the TREAT trial then was designed to fill that gap, to ask the question specifically, is it reasonably safe to go ahead and use ticagrelor in the first 24 hours after fibrinolytic therapy as part of the antithrombotic therapy? And you have to make the assumption that there would likely be benefit from doing that based on the Plato trial, that if it's safe to do it, then it's probably a good idea, based on other data, to use the more potent and consistently acting ticagrelor as your antiplatelet agent compared to clopidogrel. So the design then was to take what ended up being about 3,800 patients who'd received fibrinolytic therapy. Importantly, they needed to be less than or equal to 75 years of age, partly based on concern over risk of intracranial hemorrhage in the older population, and the fact that clopidogrel wasn't studied in this way in that older population, and then randomly assigned to ticagrelor as was given in the Plato trial, or clopidogrel as was given in most trials, beginning again within 24 hours after presentation for myocardial infarction, treated with fibrinolytic therapy. And then the primary outcome was TIMI major bleeding at this 30-day endpoint. And this was academically led. It was really Otávio who kind of is investigator-initiated, modestly funded, very efficiently conducted with a group of leaders, mostly from middle-income countries. It was open label, but with appropriate randomization, intention to treat analysis, and blinded outcome adjudication. The sample size provided power to address a non-inferiority issue. Again, this is something that's somewhat subjective. How do you define non-inferiority for bleeding? The decision here was if there was
assurance with confidence that there was less than a 1% increase, absolute increase in the risk of TIMI major bleeding, that would be evidence for non-inferiority. And then, again, using efficient trial conduct designs, Renato and I and Karen and John Alexander were involved from the DCRI, and these were the countries that were involved. The largest number of sites in terms of countries were from China, Brazil, Russia. And the key thing which we didn't fully anticipate was that it was several hours, in fact 11 hours, from the time of fibrinolytic therapy until randomization into the trial. So this was a criticism that we dealt with, is that in fact this wasn't initiation of clopidogrel versus ticagrelor in this patient population, but it was what do you continue with after the patient presents to a hospital where there's an opportunity to decide whether to continue clopidogrel or ticagrelor on average 11 hours. And most of the most common fibrinolytic agent was tenecteplase in the trial. And so most patients had already received clopidogrel with their fibrinolytic therapy. Most people had received 300 milligrams of clopidogrel, almost 87%. And then most patients ended up undergoing PCI, and adherence to study drug was reasonably good. So the primary outcome is shown in the yellow box here. TIMI major bleeding was really almost identical between the two treatment groups, and it did exclude more than about a 0.6% higher absolute rate of bleeding in the ticagrelor versus clopidogrel arm. So it met our criteria for non-inferiority between the groups. And then Plato major bleeding and Bark 3 to 5 bleeding, important secondary outcomes are also shown here. And one sees that total bleeding was increased, not surprisingly, as one would expect with the more potent antiplatelet agent, but that the most serious types of bleeding, one could argue the key type of bleeding, although we were very much underpowered to address it, was intracranial hemorrhage. That's what you really want to avoid having occur in excess with a more potent antithrombotic regimen in this setting. And there were 8 in the ticagrelor and 7 in the clopidogrel arm. So again, not very few events, but reasonably balanced. Fatal bleeding also balanced. And then when one looked at, because it was 11 hours after fibrinolytic therapy, an obvious question is, how about those patients who were randomized earlier after fibrinolytic therapy? The bleeding rates are higher there, both because ascertainment of bleeding begins earlier in the process, so you're capturing more of the early bleeding, as well as the fact that early after fibrinolytic therapy, with more potent antithrombotic therapy, one might expect a greater differential, but we didn't see that. In fact, there were also comparable rates of bleeding in the early hours in those patients who were enrolled in the early hours after fibrinolytic therapy. Clinical outcomes, again, many too few to have confidence, but they seem to be reasonably balanced, and the 1-year outcomes will be the most important to address whether or not there was a clinical benefit that outweighed any risk over the course of 1 year following STEMI treated with fibrinolytic therapy with ticagrelor. So the conclusions then are, for this population age less than or equal to 75 with STEMI, administration of ticagrelor after fibrinolytic therapy was non-inferior to clopidogrel for TIMI major bleeding at 30 days. Total bleeding was increased, and there was no difference in these exploratory efficacy outcomes, but it seems to be a reasonable option now to go ahead and treat patients with ticagrelor beginning in those first 24 hours after fibrinolytic therapy. Thanks for your attention.
Questions for Chris?
Nice presentation. Could you comment on the open label design? And certainly the endpoints are blinded and adjudicated, but when you're looking at the potential for bias related to actually reporting of the events?
Yeah, it's a good question. And I think, we kind of claim that if you do probe, blinded outcome assessment, that protects you against bias. And it does protect you against one type of bias, but not ascertainment bias. So if there are more events that are identified based on knowledge of treatment in one arm, then there still could be some bias. So I think it's still possible that there's bias. I think, our sense is that in this type of a multi-centered international trial, that there's not a major bias according to treatment assignment. But I think it's a legitimate question, and there's certainly possibility of bias. The bigger issue here, frankly, was the bigger challenge especially in a pragmatic trial looking for bleeding events, is there international differences in ascertainment of bleeding. So for example, the event rates, and I don't think we've even presented this, the event rates were quite low in Asia for bleeding, probably related to the fact that that the medical records don't document things as much. So those are issues. Presumably there wasn't a bias between treatment groups, but it is a issue in conduct of these trials.
Yeah, I think I bring it up because this pragmatic design is so important, and often we have a really hard time actually getting to the point of doing these studies because there's such aversion to having open label, but it's something we should be doing if it can be mitigated.
Yeah, and of course, the harder the outcome, the more objective the outcome can be, the less chance there is of bias. So for something like mortality, we have a lot more confidence in it and bleeding is somewhat of a subjective outcome. So it's a good point.
Another question? In the back. Yell it out.
Thank you, Dr. Granger. The question I had was, how do we translate this in clinical? The patients enrolled in this trial, had their cath, didn't have access site bleeding or didn't have any bleeding issues to be randomized into the trial. But now when we say, okay, this trial suggests that it would be safe to switch patients who've received Latebreakers to ticagrelor, it should be conditioned on patients not having had bled, to have actually met criteria for enrollment or for eligibility for randomization. So how do we translate this, in regular clinical practice? You
mean like for the question about upfront treatment?
Right.
About whether, so we've been, a good question. And I think what I would say is I would probably apply this treatment in practice, including at Duke, to say that it's probably most reasonable to go ahead and give clopidogrel with the fibrinolytic therapy and then make the decision about whether to continue with clopidogrel or ticagrelor at the next dose, 12 hours later, because we really don't know. It is possible that for patients treated with fibrinolytic therapy that there is, and again, the risk I worry about is intracranial hemorrhage. And I'm just not confident that we know that giving a much more potent antiplatelet agent at the time of fibrinolytic therapy is safe. But I think it's fine. It's a little bit like the issue of bivalirudin and heparin, like in Horizons. Patients actually did better if they got heparin beforehand and then were switched to bivalirudin when they got to the cath lab. Same general kind of thing. And I suspect that's what the guidelines would reflect, assuming they find this a compelling set of data to help change the guidelines.
So quick one, what happens if the one-year results come back and there is no difference between the two arms?
Great question. Right. Sometimes you don't want to ask a question that you don't want to know the answer, or you think you already are certain of the answer. So because we have, this is a small study. We have a much larger study showing that for a year of treatment, the outcomes were 16% relative risk reduction in the composite and reduction in mortality. So what would we do if we didn't see an effect on mortality? On clinical outcomes. I think we would probably say that that Plato is a more robust set of data to address that issue, and presumably after 24 hours or some period of time of a STEMI treated with fibrinolytic therapy, it's the same population. Patients were enrolled in Plato who were beyond 24 hours after fibrinolytic therapy. So you could even, and I think you were asking this in a provocative way, why are we even including this as a substantial focus if we kind of already know the answer and this is not a big enough trial to add meaningfully to that? But hopefully we'll at least see a trend that's consistent with Plato, and then we won't have to directly answer your question.
Sounds? He did say trend.
Okay. Comment on kind of in the post-Plato, non-North American benefit, aspirin dosing. Will you be able to tease out whether that signal that was seen in Plato in a subgroup, and whether aspirin dosing has implications here.
Well, the problem is, the only North American patients were in Canada. So, and I forget how many, a couple hundred in Canada. I mean, we have some in Canada. We mandated by protocol that the aspirin dose be less than 100 milligrams. So I don't think we'll be able to substantially address that, but it's be interesting to see.
Great. Winding us up will be Renato doing Secure.
Great. So this is a trial that actually we started discussing about 7-8 years ago in Brazil, so a long time. And at that point it was very exciting because we were trying to convince the Ministry of Health that we could actually deliver outcome large randomized trials, because traditionally in Brazil, most of the grants, most of the money goes to basic science, and trying to, with the creation of the BCRI and with the partnership with HCOR in Brazil, we started to try to prove to the Ministry of Health that we could potentially do something meaningful. And so that was the background behind the conception of the trial. And of course, we needed to think about a topic that we believe we could do it. And the background was, okay, we know that statins everybody needs to be using after an acute coronary syndrome. It's a class 1 recommendation, but we have less information about the timing. Should we give up front? Should we give in the middle of the hospitalization? Should we give it a discharge? So we had less information about when to give it. And there were some studies showing that every time you're giving a loading dose, so a high dose statins, prior to the PCI, you improve outcomes. So there are some initial studies showing that. At least improve outcomes related to the procedure, because maybe there was a pleiotropic effect of statins. Of course, this effect is not related to lowering LDL, because it's not possible to do it in 24 hours. But this was probably related with other effects of statins. But most of these studies were small, and most of the studies are in the setting of elective PCI, so patients just go for an elective procedure. So we really wanted to say, okay, we need this answer in a large pragmatic trial looking at the spectrum of ACS. So a patient presenting with the whole spectrum of ACS, ST elevation myocardial infarction, non-ST elevation myocardial infarction, unstable angina, and we give upfront, and of course, with the intention of a PCI being performed. So that was the rationale behind this study. And based on that, that's basically the design of the Secure PCI trial. Basically, we got the whole spectrum of patients with ACS. They were intended to be treated with PCI. And of course, we accounted knowing that not always everybody that got with an ACS is going to undergo PCI. So we predicted in our sample size calculation about 70% of patients getting a PCI. And then we randomized patients to atorvastatin, 80 milligrams upfront in our initial loading dose. And then after the PCI, in the next 24 hours, another loading dose versus placebo. And then after that 24 hours, everybody got the same atorvastatin, 40 milligrams, up to 30 days. So the primary endpoint was MACE at 30 days. That was adjudicated all-cause death, non-fatal MI, stroke, and unplanned revascularization. So again, looking really at the question of the initial early phase loading dose presentation, then followed by the regular statin that we usually use. And for logistic reasons, we chose 40 milligrams atorvastatin, which is considered one of the higher dose of atorvastatin. So we thought it was ethical to go with 40 milligrams. The trial has basically a Brazilian steering committee. We started 7-8 years ago with these discussions. John and Chris help reviewing the protocol. I have the pleasure to chair the trial with my friend Otávio. And we included 53 sites in Brazil. And more importantly, we were able to get the money from the Ministry of Health. So the Ministry of Health believed in the idea and helped us fund the trial. And also important, there were two coordinating centers, was the BCRI, the Brazilian Clinical Research Institute, and the HCOR, as mentioned before. In terms of the primary endpoint, so here what we can see that we didn't find any significant results. We found a 12% reduction, but not reaching statistical significance on the primary endpoint, comparing the true loading dose of atorvastatin before and after PCI versus placebo. The results were pretty much consistent according to different subgroups, with the exception, what we initially anticipated, the PCI subgroup. And we found a statistically significant interaction in the treatment effect in patients undergoing PCI, demonstrating that it seems to have the loading dose atorvastatin seems to have an effect in those patients who underwent PCI, but not in the patients that did not go under PCI. I'm looking at Karen already in the subgroup analysis, making faces here, and I'm being very careful in what I'm stating here about the subgroup, post-baseline subgroups, and all that thing. But because it is an important subgroup, and we pre-specified that subgroup because we initially actually believed that most of the effect would be in the PCI, and we anticipated 70% getting PCI, and we end up with 65%. So we were a little bit short in our rates of PCI, although not that short. We got 65%. But when you look at that, then we basically found this 28% relative risk reduction in MACE in those patients who actually receive the loading dose versus placebo. When we look at the components of this MACE, it was pretty consistent according to all the components: death, MI, stroke, peri-procedure MI, non-peri-procedure MI. And we also found some very interesting that these results were even more important in the patients that have presented with STEMI, with basically a reduction of about 50% in the rates of stent thrombosis. So again, all in the right direction despite being a post-baseline subgroup, but in the other hand, pre-specified. So the conclusion of this trial was that among patients with ACS and planned invasive management, the peri-procedure loading doses of atorvastatin did not really reduce MACE at 30 days overall. Therefore, our findings do not really support the routine use of this loading dose, at least of atorvastatin, in unselected ACS population. However, we found that these patients undergoing PCI, the loading dose seems to improve clinical outcomes and might be an attractive treatment strategy for patients with acute coronary syndrome that we know are high likely to be undergoing PCI, particularly if they present with a STEMI. And again, I think we're going to have to give statins anyway. We did not find any side effect in both groups in terms of placebo and statin. So why not maybe get up front, particularly in the patients that undergo PCI, because we might be getting 30% reduction in MACE at 30 days. We're going to be following these patients up to 1 year as well. So hopefully next ACC we might present the 1-year results, and we might get more power, more number of events, and these results might change a little bit. We were fortunate enough to publish this paper in JAMA, which was very important for us in terms of planning with the Ministry of Health, getting more funding, getting more papers, getting more trials ongoing in Brazil. Thank you.
Questions? Renato, mechanism of action. You gave 1 dose of a statin. By the time you reach day 3 or 4, you're at similar, I would imagine, lipid levels. Why in effect?
Yeah, so there's a bunch of theory, Eric, in this regard, and of course it's not LDL related. So it's people who study really the mechanism behind statins believe on the pleiotropic effect in this high really dose of statins, anti-inflammatory, particularly if you go into PCI, there's a lot of manipulation of vessel, so you might get better anti-inflammation by those high dose immediately doses and see the benefit in 30 days. So that's just the hypothesis. We cannot prove anything mechanistically speaking with this study.
Great. Chris?
So of course, Renato, I totally agree with your interpretation.
I'm glad.
It was peripherally involved with helping to formulate it. But let me ask this question, including to our statisticians. When you see in the group that didn't, so you have an overall trial there's a trend, a little bit of a trend for a better outcome in one group, and then you do a subgroup analysis. And in one of the groups, you see a 36% point estimate of a 36% worse outcome. and the other group at 30% better, like, do you have to also conclude that there was harm in patients not undergoing PCI, or does the fact that it was a 36% point estimate of a higher outcome, but the confidence intervals were wide, does that get us off the hook? Or how do we, how do we, like, you know what I'm asking? If overall there wasn't a significant difference, then how tricky is it to interpret that one group really had benefit?
Okay, so, I just want a point of clarification because I saw a p-value 0.27. Is that right?
For the
So
primary, yeah.
for the Indy trial, our p-value was 0.27, and I don't recall talking about trends, Chris, or subgroups, but
It's in the eyes of the beholder.
Okay, this is an opportunity for the audience to ask any or all questions. I think the first thing that I would point out is at least 3 out of the 4 of these presentations presented results that were done in the trials were not fully traditional styles, and they were very pragmatic. Two of the trials done mostly outside of the U.S. In countries that are considered more developing or mid-tier countries, I think is saying a lot. And then Tracy's being done in the U.S. but being done in a highly pragmatic cluster design. Discussion about amongst you guys?
So I'll just say, Eric, that again, when we created the BCR, for example, the idea was to bring the knowledge that we have here at Duke to there, to be able to maybe cheaper and in a more pragmatic way, in a less complex way that we do in the US, but keeping the same scientific rigor. And I think those type of initiatives is our job now to really, it's clear that we need to move out of the US to be able to complete this 10,000, 20,000 patient trial, but the challenge for us is how to keep the quality, how to make sure that we don't get underreporting of events, the ascertainment bias is not a major issue, and how can we then defend the results, being certain that the results will have been the same should the trial have been done here at the DCRI. I think that's kind of what we're working, at least part of my life that I'm dedicating to.
Yeah, and I'll just comment. I think it also shows, doesn't it, nicely, both the strengths and the limitations. I mean, the pragmatic, these trials wouldn't have been able to be done because they didn't have, at least the two from Brazil, there wasn't a huge amount of funding, so they had to be done in a pragmatic way with networks that are less expensive to do these types of trials. And I think in some ways then they're also more applicable to middle-income countries and probably maybe a little bit more generalizable because of some of the design features. But they're also were a little bit smaller, right? Both of them would have been, I wish we could have had more resources to do larger trials because as we've been talking about, the results have wide confidence intervals. There's a little bit of uncertainty as to the robustness of the nature of the treatment effects.
I just was curious, hearing Tracy and her experience here with Artemis, as we're thinking more and more of leveraging systems and cluster randomized trials. I mean, there are several that are ongoing now that DCRI is leading and or developing. What's the key things to not do or to do? We need this now for other studies.
Yeah, I think this has really been, for us, a bit of a learning curve, if you will, because I think there are definitely lots of advantages to think about these pragmatic trials. First of all, you may not necessarily need industry funding because these would be, allow you to answer questions that perhaps industry in and of itself is not interested in answering, so being able to sort of broaden the scope of the research questions. In part because we know that, all the clinical trials that we do, they represent less than 1% of the of the population of patients that we treat. So really being able to make clinical trials easier so that we can actually reflect the rest of the 99% of the population we treat is really important. So I think there's definitely a lot of strengths to be wanting to do these pragmatic trials. I think from an operational standpoint, I think many in the room can attest to this. There are also unique challenges to doing pragmatic trials. one of which is simply communicating exactly how to do this trial to people and what it means to be part of a pragmatic trial. And then the other is making sure that we have a set of standards for how we do the research and whether or not, you know, those set of standards is compromising the science in any way. And we'd like to think no. We think we can get there without compromising the quality of the data that we're trying to get here. So I think for many of us, I think every trial is obviously very unique with its lessons learned, but I'm pretty excited about Artemis because this is actually one of the largest pragmatic trials there is out there that, you know, is purely done in the U.S., which is, as many of you know, one of the hardest places to do clinical research worldwide now. And so the fact that we were able to make this experience work, we were able to get very good follow-up on the trial patients that we enrolled into the trial. We were able to do that from a centralized location without necessarily requiring the patient to go back to where they were enrolled. We were able to, you know, follow patients longitudinally with very little loss of follow-up. All of these, I think, are lessons learned and can be propagated as we think about doing other trials.
Yeah, I think the only thing I'd add to that on Artemis, at least, it was the issue of the cluster randomization itself was interesting. You know, enrolling institutions in a study that looked at giving away free med was actually, you know, easy. That was the great part. Everybody wanted to sign up, and we had no problem getting sites signing up. And among those in the intervention arm, we had no problem enrolling patients. The enrollment went incredibly well, much even ahead of where we anticipated. The challenge was obviously the control arm. Your center signed up for a study, and now nothing changes. And at a cluster level, that's pretty hard to deal with, and we saw it. Sites didn't enroll as fast or anywhere near as fast in the control arm. We even added in extra control sites, which may or may not exactly be perfect, but we worked it out. It didn't affect the overall results of the study, but we still could never catch up. So figuring out ways in which we could design studies better in the future that might have more incentives, you know, either a 2x2 design or other ways in which you can potentially test this without having each side feel like they're getting some benefit for participation. But it was clearly obvious in terms of the enrollment issues.
Yeah, I agree. I think that's a key issue. The other thing, Tracy, you were talking about this yesterday, and I think it's so important, is the issue of consent and the fact that simply getting consent from patients, you know, it's a huge barrier, right, from a truly pragmatic study where we represent true unselected populations. And we now have two trials that we're doing without consent, which is actually kind of interesting, I think. Both with Sean Picorni's involved with both of them. One is the FDA Sentinel project, and the other is a project, Quantum AF project, and then we're proposing a third trial, a cluster randomized trial of counties in North Carolina to improve cardiac arrest care with no patient consent.
Waiver.
Well, yeah, it's waived, but the result is a totally unselected population. Thank you.
Cover.
With appropriate.
Mrs. Smith, would you like CPR?
Achievement of waiver of consent based on OHRP criteria for waiver of consent. And careful review. I mean, in fact, the FDA sponsored trial, we had ethicists from the FDA and the NIH weigh in because there were obvious questions about when is this appropriate and is this an example? And it was determined at the highest level of ethical review from government, regulatory, and funding bodies that this was an example of that. So I think it'll be interesting to see how it plays out.
Never is our government unethical. Just remember that. Karen.
Can you guys please define for us pragmatic trials? Because you've said that 3 of them are, and I'm assuming the 2 from Brazil are the other 2 that you're considering to be a pragmatic trial. But it felt like a standard trial to me, both of them, in what the fact that it was in Brazil and possibly less expensive. But I mean, it felt like it was patient randomization. It was, in most ways, it was very similar to the trials that we do here that are considered more standard.
Yeah, I think that's a topic even for a broader discussion, how to define pragmatic trials. And I'm meeting another day, and there's 5 people in the audience 5 people in the panel, and 5 people have different concepts of what pragmatic trials might mean. I think what we meant by that is with less complexity, much easier to get approvals and faster, much cheaper. The monitor was very limited and was based on really...
Can you share?
Only that.
Are you able to share?
Yeah, we did a trial for $1.5 million. For 4,200 patients.
1.5?
Yes.
How about treat?
Treat was about $10 to $12 million.
You're killing me. Okay. We're all going to Brazil.
So yeah, so I think that's part of the thing. You have to streamline. So adjudication, I mean, we did everything we did, but we rely on the investigator initially starting the process, as opposed to us developing complex triggers, for example, to trigger peri-PCI-MI based on biomarkers, because we didn't have the money to actually have biomarkers every 3 hours to try to pick up increase or decrease or trends in biomarkers. And there is a price to pay to that, because there is likely missing events. There are likely events that we were not able to capture because we were less rigorous in the way you do, but there is no magic. $1.5 million to randomize 4,200 patients and get a paper in JAMA. I think that there's so much one can do, but that's the piece that that I think we should focus on. How can we maybe not get to the $100 million, but maybe get to the $10, $20 million and do extremely high quality as if it was here in the US.
Let me just challenge Karen a little bit about the pragmatic trial. So like you said that this is kind of our standard trial. So I know...
It sounded like it from what I heard.
Yeah. So here, maybe this could illustrate that. So like so-called pragmatic trials that we've done in the past have been more pragmatic than the others, but it still generates a CSR that's like this tall. Whereas like, it sounds like from what Renaud's describing for the 2 studies in Brazil, like there's not a CSR that's like, measured in feet, but actually it's very simple to the objectives and focus data collection, for instance, and all the procedures. So that at least in terms of population, the type of data, the intervention, and being more embedded in the real world seems like key features that you were describing that are different than, say, our standard pragmatic trial.
I mean, there is the scale, right? I forget who created it. This like Prism 2, yeah, like 10 dimensions, and you can graph out where your trial fits on that, and that's, I don't know, it's far from perfect, but it does give some idea about the elements that many people think define how pragmatic a study is.
So we're at the top of the hour. I'd like to, in part, recognize all those who were both presenting the results to us today, but also to all of you out there who helped carry out this work. These studies are answering important questions. They're doing it in new and innovative fashions, and hopefully they're just the beginning of where DCRI is going in the future. Next week, just one final announcement. The guest lecture had to be delayed for family reasons, but we will have Adam DeVore here, perhaps with something. There'll be Get With It guidelines and a few other things associated. So looking forward to seeing you all here next week. Thanks.